This is TWIS. This Week in Science, episode number 915, recorded on Wednesday, March 1st, 2023. What's your science superpower? Hey everyone, I am Dr. Kiki, and tonight on the show, we will fill your heads with brainy computers, urination superpowers, and lots of old people. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. With each test and trial, science finds new ways to thrive, pushing past boundaries. Through the microscope, science sees wonders of life, large yet very small. Inside every cell, DNA holds life's blueprint. Science reads its code. Stars light up the void, cosmic darkness all around. Science will explore. Clever problems solved, animal minds amazing. Blair has got the proof. Within our own skulls, the brain's electric light show. Kiki points the way. Ancient bones and tools, the past revealed under earth. Justin brings to light. This week's disclaimer, somehow stuck in haiku mode. The show won't be so. Sit back and relax because This Week in Science is coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. We have so much science for you today. As every Wednesday, we like to bring you our science news stories that we think are fun to talk about. Thank you for joining us. Hopefully you'll enjoy the show ahead. I have stories about brains, more brains, and hauling logs, and also an unexpected insect at Walmart. What do you have, Justin?
Oh my gosh. I've got an ancient platypus of Patagonia, meandering Iranian Neanderthals, and a couple of stories out of Max Planck Institute about the origins of current modern humans in Europe.
More origin stories. We do like the origin stories. Yes. Superhero power origin stories. Blair, what's in the animal corner?
Well, I see that Kiki, you have stuck to your wheelhouse very, very strictly this week. Justin, also, I have as well in the animal corner this week. I have urine and hormones, so it's very safe territory for me. Also, I'm going to do a quick story about Norse sea beasts and octopus brains, just for fun at the start of the show.
Yeah, octopuses and wee beasties. The Norse version.
Big.
Right.
I've got three guesses of what the Norse sea beast could be.
Well, you'll hear in a second.
Yeah. Okay.
I'm getting all squiddly about it. As we jump into the show, I would like you all to remember that if you have not yet subscribed, you can subscribe to us. All places podcasts are found. Look for This Week in Science. We are also streaming weekly live Wednesdays, 8 p.m.-ish Pacific time on YouTube, Facebook, and Twitch. We are TWIS Science on Twitch, Instagram, and Twitter. And you just go to our website, twis.org, if you have other questions about things, show notes, and all that. But it's time for the science. So let's do it. Who wants to talk about brain computers?
Wait.
So is this going to be a computer that goes in my brain, or a computer made out of brain?
Right. Yeah.
Is this a big
It's a big difference, right? Yeah, huge difference. This is a computer that researchers in the Disseroth lab have just published their roadmap for in Frontiers in Science that they're calling organoid intelligence. These are little brain organoids that could be used as computers that could be connected together to create neural networks, working computers made out of neurons, biological materials.
Oh, cool.
This is
It
Johnny
is.
Mnemonic, is what this is.
It's a fascinating idea, and we've been talking about the concept of brain organoids and what we're using brain organoids for. And the basic idea is that we can use brain organoids, which are several thousand, maybe 50,000 cells or so, very small little models of the brain, so that we can look at developmental issues, we can look at disease, we can start figuring out how components of the brain work together. What they're wanting to do is, I mean, you got to scale up to like 10 million cells per organoid, which is not a small deal, but they want to make bigger organoids, bigger, better, enrich them with cells and genes for learning, make sure that they're able to remove waste, because that's very important for the living creatures, and also have precise chemical signaling. They have basically created this idea of three-dimensional cultures of cells where there is biological hardware and wires that connect them together, so that these neurons, these little organoids, could work together and create larger computers. The current researchers who are working on this have said, actually, I said this was the Disarath lab, and that's actually a study that is later that I was going to talk about in the show. This is actually out of Johns Hopkins University, Professor Thomas Hartung, his lab. And they're thinking that they might make good computers, because the researchers say, well, silicon-based computers are certainly better with numbers. Brains are better at learning. So the learning aspect is superior to the way that computers learn at this point in time. And so from an energy perspective, it might be better for information storage and learning. And we are also reaching the limitations of where silicon chips are going to be able to take us.
Yeah, but
Okay,
we've
so
always
we're
been
trying
reaching
to-
that. We've always been reaching that limit. Haven't we always been reaching that limit? And then a new computer comes out and we're like, oh, look, the limit's totally different now. And
This
True.
one
oh,
has
we're
16 cores.
True, but so Moore's law of computing is that, you know, the transistor is getting smaller and smaller and smaller, and so we actually are reaching, basically are at that point where the atomic limit of like the size of an electron hopping from one part of the transistor to another, we are reaching that limit. And if we want things to get smaller, components of computers to get smaller, so that we can have faster, bigger, better, stronger computers, we need different materials. And so there are wide-ranging efforts in this arena trying to figure that out. But it's fascinating what they're thinking about doing here, though. They want to adapt tools from like bioengineering and machine learning. They want to engineer new stimulation recording stuff. They've developed like a little tiny mini EEG cap for little brain organoids, so that they can use electrodes to pick up the brain organoid signals, similar to the way that we use EEGs to pick up signals from the surface of brains today.
So here's my problem with this. Without opening a whole can of worms here.
Oh, I opened it already. Worms everywhere.
That's right. So machine learning. We are now going to enact that in a biological living entity. So now you are trying to basically engineer your way towards AI in something that is alive. This is problematic.
Is a neuron, is alive. Yes.
It's certainly more alive than a computer chip based on our current standards. So this is very interesting territory, because this whole like, is AI alive conversation becomes more complicated when you're dealing with organic matter.
As specifically human brain cells.
Yes.
Yeah. So
it's funny because then also you're saying, Normally we might define life as something that is intelligent. And then here we would say, oh, it's only intelligent.
Right.
And
Right.
has biological, but the rest of it doesn't have, like, wait a second.
Well,
So first of all, I'm okay with avoiding this and just having computers double in size every few years
same.
because
Yeah,
they're
let's
small
just-
enough.
let's
We
just-
can have much
It's
bigger
fine.
computers. That part's fine.
I am all for having like the 1980s boombox cell
Yeah.
phone. That'll be great.
Yeah, it'd be easier to find.
Yeah.
The thing though is too, this changes that sort of cyberpunk future of humans adapting and having a
being
technology added
cyborgs
to
and
them
stuff, yeah.
piece by piece over time to integrate with machines. This route, the future cyborgs are going to be biological tissue based again. With
Yeah.
just it's going to go the other way. It's going to
Yeah.
slowly be adding more biology instead of more tech.
Right. And so then you start getting into ethical issues. And so the researchers not always, they also took into account the fact that they need to be talking to all sorts of stakeholders in this situation. They're going to need to be taking the public, ethicists, scientists, all sorts of politicians. People are going to have to be taken into account in the conversations about all of this as we go forward. Human little brain organoids. One little organoid with 50,000 cells, not a real brain. Fine, no consciousness, whatever. But what if you connect them all? At what point
Right.
is consciousness developed? Does it develop consciousness? Can we measure that? And then if it does have consciousness, can we model Alzheimer's disease in it? And is that okay?
Right. Oh no.
Yeah, so there are a lot of very interesting philosophical questions, ethical questions that need to be asked and considered as these researchers are pushing toward developing a community of researchers who would work in this direction. And on that note, let's move forward to something else, like ancient Platypus agonia.
So this story, I kind of at first had real reservations. Could they really have found what they say they found? So, because all they found so far is a tooth dating.
Sure.
It's found down in Patagonia, is the southernmost region of Argentina, in this formation where they've also found dinosaurs, sauropods and theropods, snakes, turtles, frogs, mollusks. This was a very different climate 70 to 60 something-ish million years ago. And they found this one tooth that they looked at and say the morphology of it clearly belongs to a monotreme. More specifically, they think it's a platypus tooth. Now, a couple of things there. First of all, as Blair might know right away, platypuses don't have teeth. So there's one problem. The other thing is, this is the southern tip of South America. Platypuses only exist in like Australia.
Australia.
That's it. So did they just like, oh, we couldn't identify this tooth from the creature that we don't know what it is?
Well, so
And-
okay, hold on. So they currently are only found in Australia. But if you prescribe to the previous explanation, monotremes are the ancestral mammal. And so in theory, you could have had monotremes all over the world that then later developed a placenta. So that's possible. However, I did bring a story a few weeks ago that ruined that entire theory, potentially. So I don't know.
Okay, so here's what's kind of going on. First of all, turns out there was another platypus discovery in Patagonia, or in Argentina at least. That study 30 years ago also only found a tooth. So then I'm thinking, well, maybe they couldn't identify it, but because somebody else had identified a tooth as being an ancient platypus tooth. Maybe that was it. And it turns out ancient platypuses did have teeth. The current ones actually do too when they're young, but they lose them. Which is they lose them and they get replaced by pads when they're juveniles. Okay. So, all right. And the morphology of this tooth is so specifically different than other things that, yeah. Okay, maybe. The other thing is, you're talking about the ancient ones, the ancient egg-laying mammals.
Yeah.
That stopped like 160 million years ago.
Yeah.
So this is at 70 million years ago, and the older find, the 30-year-ago find, was 62 or less million years ago. These are much more recent than that. The thing is, at that time, 70 million years ago, Southern Australia and Southern America were both much further south and connected to the landmass of Antarctica. And fun fact that I didn't know before reading this story, all of the marsupials of Australia, the place known for the evolution of marsupials, they all came from South America. That's their origin.
It's what I said before too. There were marsupials everywhere before.
Yeah. Oh, there still are. Apparently, there's still like 100 species or more of
possums
in South
with
America.
no O in front.
Without the O. But yeah, if you look at the map, like our standard map, it's like, whoa, there's thousands and thousands of miles of open ocean. But if you look at a globe from 70 million years ago and turn it to, I guess, upside down, depending on where you're standing, you see that there's a land bridge from Australia to South America, basically, that allowed animals to go back and forth. So, yeah, so pretty exciting. They, again, still only have found 2 teeth.
Yeah, I have trouble with
from
that.
2 different specimens. So they really need more finds to flesh out this fossil, to see what this creature even looked like.
Yeah, not enough teeth to really draw conclusions, but maybe enough to make it interesting.
But thankfully, though, what they're saying too is the other interesting side sort of piece of this is looking through tooth morphology of ancient platypus. The morphology of their teeth changed very little over tens of millions of years. Most mammals, their teeth evolve pretty rapidly. I mean, our own tooth evolution between days of Homo erectus or even Neanderthal compared to human, or Neanderthal within Neanderthal, changed more dramatically than a 60 million year stretch of platypus evolution. So their teeth are very conserved, which was part of what allowed them to led them to the conclusion initially that these would be connected.
Okay. Well, if you eat bugs, I guess to a certain extent, you can only improve on the teeth for eating bugs so much. Just need squishers.
I'm still a very, now that we're in the age of genomics and being able to look at DNA, the phylogenetic just morphology approach always seems like they need a little bit more.
Yep.
When it's the best thing you got, it's the best thing you got.
It's the best you got. Yes, exactly.
And then of course you're right. DNA is the best thing that we've got.
Yeah.
So if we can get it from a tooth like this, that would be terrific. 70 million years old. I don't think there's going to be a way to do that yet. But yeah.
Not yet.
You're right.
But
that would answer many questions.
So many. Blair, tell me about some ancient sea creatures, please.
Yes, this isn't 70 million years ago. This is maybe 2,000 years ago. But this is all related to something actually found in 2011, which is scientists recorded a really weird feeding strategy in whales around the world. Whales are known for lunging at their prey when they feed. But in 2011, whales were spotted at the surface of the water with their jaws open at a right angle, waiting for fish to swim into their mouths, for thinking that their mouth was just a place to shelter from predators, but actually chomp. And so scientists speculate this is either the result of changing environmental conditions, or
They
that
were
we're just-
hybrid with a Venus flytrap.
Good idea.
Yeah, no.
Or that whales are just being watched more closely by drones, by other modern technologies. And so we're just catching this behavior, which we couldn't really catch before, which is probably what's happening. So wondering if this is a recent phenomenon or if this is something that we just haven't seen but has been going on for thousands of years. I don't know. Dr. John McCarthy, a maritime archaeologist, in the College of Humanities, Arts, and Social Sciences at Flinders University, noticed interesting parallels between this phenomenon and some historical literature about Norse sea monsters. So in the Norse description of the Hafgufa, they had a very similar behavior. They were, it seemed to be just like these whale traps. At first, it was thought to be an interesting coincidence, but once you looked closer into the detail of the description of the Hafgufa, and once he discussed it with colleagues who specialize in medieval literature, they realized that the oldest versions of these myths most likely described whales.
Mm-hmm.
So, these Old Norse manuscripts describing this creature from the 13th century alongside other myths up until the 18th century, including kraken and mermaids, they were doing this strange behavior. So, they actually think that this could be a clue that whales have been doing this for thousands of years. So it's a really interesting use of mythology, ancient mythology, to describe an actual biological phenomenon. This is pretty cool. And so this has a potential answer to the question of how long whales have been using this strange technique. And
Forever.
obviously marine biologists thought there was no way to figure this out. But medieval manuscripts about monsters had a clue.
I like the images you were sharing there, Kiki. You can sort of see the game of telephone that takes place from between perhaps illiterate sailor and an artist rendition later, getting a verbal explanation of the half-glimpsed creature that was seen in the water, and then trying to do that artist recreation based on that limited information and a sort of pre-existing knowledge of what animal parts should look like. It's pretty amazing.
Well, it's also, the not having all the information that we have now about how things happen under the surface of the water and the fear of what's under the water, yet people sailing on top of it. And so only knowing what they see from the top or what they drag up from below in nets or on fishing poles. Or these, like you said, these tales that the sailors bring to port while they tell while they're drinking or what have you. Yeah.
Imagine if you only saw that first video Kiki shared of just a giant mouth. You did not see any body. You just saw a giant mouth. And then you think about maybe like 100 feet away, there's another whale cresting. Then you're like, oh my God, it's a sea monster. It's fully a sea monster. So you could see how to Kiki's point, thinking about just the surface, what you just see on the surface, you could totally kind of manufacture this phenomenon. But I love using this kind of cryptozoology lens to actually help with real science.
We have all sorts of evidence like that going back years. It's just a matter of figuring out how to put the pieces together, right?
Absolutely.
Takes people.
Yeah, and this is there is another example of this in some cave art that was of a bison, and the bison didn't fit any existing bison. So it's like, artists, crude attempt at drawing a bison. It made the rump too big or the shoulder too pronounced or whatever it was. And then they discovered, actually, this was in a channel in the Ice Age where there was two separate groups of bison, and they discovered a hybrid between the two that looked exactly like the cave art. Yeah. So, the ancient peoples, they were actually seeing the real world from their perspective back when they were seeing it. So we should be paying more attention to ancient stories like this, because there's probably more truth to them than we actually think.
Absolutely.
Lots of truth. More ancient stuff, except I bet you didn't think you could go get it at Walmart.
What?
No.
So an interesting story out this week, published in Proceedings of the Entomological Society of Washington. Researchers have been talking about a find of the researcher Michael Skvarla. He's the director of Penn State's Insect Identification Lab. He was at a Walmart in Fayetteville, Arkansas. Saw what he looked like, a lacewing insect kind of thing. Was just like, oh, he didn't, I don't know. He's like, oh, what is this? This doesn't look great. And he grabbed the insect and he said he cupped it in his hand and did his Walmart shopping and then took the insect home as well. And
no barcode on that.
No barcode on this this insect. But the story is really fascinating because he found this insect back in 2012, held on to it, but then COVID happened. The pandemic put all sorts of researchers as instructors online, doing things that they would normally not be doing. So he took this insect to class through Zoom and did basically a demonstration to look at the characteristics of what he thought was an antlion, but then started going through the characteristics through this Zoom class and
live. I love
it. And his students, and he realized it's totally mislabeled. Wow. That this was for the first time in maybe since the 1950s, the identification of what's called a giant lacewing, Polystoicotes punctata. It used to be all over North America, but it really, really hasn't been, and it hasn't been around since the 1950s. They think that the populations might be relics of the Jurassic era. It's just a very interesting point that the researchers are wondering now, is this a discovery? How the heck did it get here? Is this just random insect that showed up with bananas at Walmart or, whatever? Or is this recovery, and is climate change and all sorts of other stuff that's going on impacting the environment that this insect can live in?
It could be, but then it's not going to bring it back. Right. That thing's been surviving somewhere unseen all this time.
Somewhere. I mean, it's been around. So it used to, according to this article, used to span from Alaska to Panama. Multiple ecosystems, regions, eastern and western North America. But this is the first time.
Now survives on aisle 6.
That's right, in the Ozark Mountains. So it's they're saying this is where it was found. It's possible that it could have just been hiding and populations dwindled and nobody's really noticed it. Mm-hmm. But it's also possible that they've started recovering. And so this is the big question that we don't know the answer to.
Yeah, I think it's much more likely that it's
a hitchhiker,
a combination of a lack of the species being present and a very small percentage of the population that are entomologists. Now, those two together.
Over. Yes.
Yeah.
Yeah. So interesting. Walmart. Go to Walmart. find your rare insects.
Huh.
Or go to Europe. What do we have to go to Europe for, Justin?
Oh, we got to go to Europe, all because that's where all the action is. If you're looking for Neanderthals, this study is researchers took. I kind of like this study because they use a lot of open source data systems. Researchers were modeling the Neanderthal migration from Europe into Central Asia and southern Siberia and Russia, where there are known sites of Neanderthal occupation that have also been genomically linked to having their origin in Europe. So they know they ended up over here in Central Asia and southern Siberia, but they don't know how precisely they got there. So this modeling, starting in the Caucasus with some groups that they believe are linked to those other, basically took climate data and created what is called a least cost path for that migration. So least cost path kind of works like your car's GPS. Your car's GPS is going to find the least cost path to get you from point A to your destination, right? Now, it's not going to divert you off-road over the mountain and, across a river without a bridge. It's going to keep to the roads because there's less cost in driving that way. It's also going to route you around any kind of construction or heavy traffic, because in the model of your car's GPS, the cost is weighted by time. So the quickest way it can get you there is the path it's going to choose. In the modeling that they did, the cost is weighted in resource availability, how that's affected by climate, how sustainable a region is for a living creature. And what they came up with is that path would have taken the Neanderthals below the Caspian Sea in this southern Caspian corridor into what is northern Iran. And what was interesting is they found that this region actually would have been not just the best path to keep walking through, but would have been really ideal to settle in. And it's an area that isn't that well archaeologically explored. So they're kind of suggesting, hey, this model, that we did trying to figure out the what the best migration path might be, could be a very important spot to go start looking very intensely for evidence of ancient hominins. Because aside from being a neat way to get out of Europe and over into Central Asia, it's also the known and proposed and ideal location to move into Europe from Africa via the Levant. So we got this region that looks like it would have been a really intense hotspot, but we don't have the fossil record there yet because we haven't done a whole lot of.
So when we going, I'm gonna grab my brushes and my shovel. Let's get over there.
I'm ready. And according to the researchers there too, they're saying pointing out that there's lots of cave systems. Like this is an area that, aside from having, one of the things the model looked at is for stable climate over long periods of time. Aside from having pretty stable climate, it also is an area that has a lot of cave systems, which the early hominins, that was the free housing for the early hominins. Hominins loved them some caves.
Give me
so
those
we need
caves.
spelunkers. We need some cave spelunkers and field archaeologists to go out and start surveying southern Caspian Sea corridor of northern Iran, because it looks like, according to this, there could really be some interesting finds there. It's also, I think, it might even butt up against Denisovans to the east. So this really might be
a hot spot,
a good mingling spot as well.
Yeah.
So there could be, this could be a source of some of the Neanderthal, Denisovan, current modern human admixtures. Just find it.
Right. If there are people going back and forth and back and forth and back and forth, they're going to run into each other, right? A lot of mixing, trading.
Back and forth. And again, as they're pointing out, a perfect place to settle. Where are you going? You're here. You have everything that an early hominin needs to have a good life right here in this location. Yeah, there are still areas of the world that have not been archaeologically explored well enough. And this points to one of those things that we've mentioned on this show a few times: more research needed.
Lots more research needed in this particular area. So how many Colorado University Boulder researchers does it take to carry a log?
Depends how big the log.
Or and how big the researcher.
Yeah.
And how big the researcher. Well, some researchers, again, using their time during the COVID-19 pandemic to their best advantage. They've just studied, they've just published their paper describing their experiment, trying to figure out how people may have carried big giant logs, 200,000 big timbers over 60 miles to a place in the Southwest that is called Chaco Canyon. And so the question is, there's no big trees in this canyon, yet they built things with big trees. How'd they do this? How did they get them there? Well, it looks like they used their heads. Literally. The researchers have devised a wonderful device that allowed them to use their heads to share the weight of carrying logs long distances. They used head straps that could be attached to logs and then use the support of the spine, the head and the spine, to be able to take the weight of these large timbers and allow individuals to move very long distances. The researchers say that this makes sense because of additional evidence that is local to the region in the way that locals carry things around the area. They used all sorts of suggestions, archaeological evidence of ancient peoples in the Southwest, what they called tump lines that are woven from yucca plants that are used to transport everyday items. But yeah, so anyway, big project, researchers having fun. They carried big logs on their heads.
My neck hurts looking at it.
Yeah, but as a follow-up study, some chiropractors at Boulder University have found that you shouldn't carry a log with your head.
Well, they discovered that it was better to carry it with their heads. They took the idea of these tump lines, which are depicted in these ceramic statues from the canyon area. They tried carrying logs on their shoulders, and quote from the researchers, it was just debilitating. It's just a dumb way to carry a heavy object.
Wow.
So,
okay.
Using their heads, they were able to train, and as they trained, they were able to carry heavier and heavier loads. They were able to carry them longer distances, and they think that this is a very possible way that they used these tump lines or these head straps connected to big logs to carry them long distances. Yeah. They use their heads. Once upon a time. But speaking of heads, Blair,
Yes.
let's talk about the octopuses.
Yes, their heads. I mean, brains.
Brains? Yes.
Yeah.
Yes.
Their whole body's their head. That's what gastropod means. Head, foot, or nope. Stomach, foot.
Stomach, foot.
I don't know. Anyway, I thought I was onto something there. Anyway, this is a study mostly about proof of concept for a new type of mechanism to study brainwaves. This is a way to measure octopus brainwaves on the move, and is a critical step forward in determining how octopus' brains control their behavior, and could provide clues to the common principles needed for intelligence and cognition to occur. Octopuses are a great study animal to look at brainwaves because they have a pretty big brain for the size of their body. They have advanced cognitive abilities, as we've talked about many times. They're super smart. And their brains and cognitive abilities have, as far as we can tell, developed independently and differently from the majority of intelligent animals we look at, vertebrates, right? But measuring the brainwaves of octopus is really hard for a few reasons. One, they're underwater. So a lot of the ways we study brainwaves involves electricity. That doesn't work too well. Two, they're soft-bodied, and they can squeeze through a teeny tiny hole, basically anything that is their brain size or bigger. So you can't really attach something hard to their body. They also have no skull to anchor the recording equipment onto. And they have these very powerful, ultra-flexible arms which can reach literally anywhere on their body. So if you put something on the outside, they rip it off immediately.
Nope, don't like that.
Gosh.
So they had to figure out a way.
Too bad they couldn't get the study done.
Yeah.
Well,
what
else we got?
No, they had to figure out a way to, of course, implant it internally. So they used small, lightweight data loggers that had previously been used to record brain activity of birds during flight. They modified them to make them waterproof. Problem one. They attached it to 12-hour batteries, so they don't have to worry about sourcing electricity to it. So that's part of problem one also. But then they also were able to attach it internally in a way that the octopuses could not rip off. So they did the day octopus, Octopus cyanea, which is a larger octopus. So it's definitely a better choice for this because you don't have to make things quite so miniaturized. They anesthetized 3 octopuses. They implanted the logger into a cavity in the muscle wall of their mantle. That's basically their big floppy head part. And the scientists then implanted the electrodes into an area of the octopus's brain called the vertical lobe and median superior frontal lobe, which is the most accessible. That brain region, they believe, is used for learning and memory in octopuses. So doing that, then they were able to return the octopuses to their home tank, monitored them by video. They woke up after about 5 minutes from anesthesia. They recovered. They spent the next 12 hours sleeping, eating, and moving around in their tank. They were not tested. They were not asked to do anything in particular. They just wanted to see, again, this is more of a proof of concept. They wanted to see if they could record brain activity as these octopuses moved around. Then, after 12 hours, they were able to remove the logger and electrodes from the octopuses, and then they could look at the data and synchronize it with the video. They identified several distinct patterns of brain activity. Some looked very similar in size and shape to those seen in mammals, but others were long, slow oscillations that we've never seen before, which makes sense. Again, they have a completely different brain. Yeah. It's shaped like a donut, people. It's completely different. Everything is different. Anyway. So they haven't been able to assign this brain activity to specific behaviors because they didn't ask them to do anything. And because the area of the brain they were recording, again, they think it is part of learning. So they would have to have asked it to do something in relation to learning and memory to try to figure out what the actual activity meant. But really, this is just, can we do this? And so they were able to record brain activity on the move. They think they could use this in other octopus species. And they want to answer questions about octopus cognition, how they learn, how they socialize, how they control the movement of their body and their 8 unique arms. And now they have a means by which they can look at the brain while specific tasks are happening underwater.
This is so exciting.
Yeah.
Yeah.
So no
this
exciting scientific findings yet.
No,
but it's
but
just
I don't know. I'm
just
gonna go ahead
doing
and run
this.
with the long, slow oscillations as being octopus is doing really deep thinking.
Oh yeah.
Could be.
Could be.
No,
Could be
And
what
they're
they've got
getting to
going on
be
when they're
the cypherpunks
camouflaging.
before we are. Look at
so much
this.
to learn.
Yeah.
So much to learn. It's so exciting. I mean, the fact that they've been able to solve a whole bunch of the problems that you mentioned, that we're able now, I mean, underwater.
Yes.
Something
Underwater
that's
in an animal that's famous for escaping things. Yes.
And being flexible and being able to get rid of things and being smart and being able to be like, I don't like what you're doing. I'm going to do my own thing.
Yes.
All of these
But
problems.
I wonder though if not having the skull in the way actually is a benefit for doing the brain reading.
Mm-hmm. I could see that.
Yeah.
A little
Put
bit.
the electrodes right in there without a saw. Yes.
Yeah.
Oh no. All right. Well, no sawing on and on and on here. This is This Week in Science. Thank you so much for joining us for another episode of our Science Talk Show. We love your support. And if you are able to support us, we would hope that you would head over right now to TWIS.org and click on our Patreon link. Patreon is how we are supported by you, our listeners. Our $15 a month and above level, you'll get a special sticker package. One new sticker every quarter of the year. There's also our $10 and more per month. We thank you by name at the end of the show. Anyone over that, we thank you by name. We love to thank you. Thank you, thank you, thank you. But really, thank you all. We cannot do this without you. All right, coming on back for more This Week in Science with that part of the show that, wait, weren't we just doing this part of the show? I think we're going to do some more Blair's Octopus Hour. No, Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Do you ever think about how insects pee?
Nope.
Never.
Well, you're gonna now. That is because an assistant professor in the School of Chemical and Biomolecular Engineering at the Georgia Institute of Technology, Saad Bamla, was in his backyard and he noticed an insect urinating. What was interesting?
Wait.
Mm-hmm.
How do you notice an insect urinating? I mean, normally they're just there.
Look very closely, I imagine.
They're chomping on
Yeah,
leaves.
Maybe he was taking a photo with his macro lens. I'm not sure. But if you look close enough, you can see, I mean, they drink, so they definitely pee. So he noticed an insect urinating. What was strange was, if you want to picture an insect urinating, you'd probably picture it just like any other animal, a stream. But in this case, this insect formed a perfectly round droplet on its tail and launched it away so quickly that it seemed to disappear like a catapult. And it did that for hours, just over and over and over.
I think we learned the dynamics of water droplets in the bug realm from what is it, Bug's Life?
Oh right, yes, yeah, they would drink it. Well, we've also talked about spiders that wear water hats, right? I feel like we've talked about that on the show. But anyway, this is a very interesting phenomenon.
Is so cool. The video that was just played,
so cool,
of basically like a water droplet, kind of like a water balloon, but without the water balloon because you don't need it because you're a bug. Creating the water droplet on a little hindquarter flipper, and then once it's formed, just sending it flying.
Yes.
Boo!
Yes.
Out of here.
Yeah, so this
You're outta here.
is a glassy winged sharpshooter is the name of the bug.
I don't think we like those in California.
Correct. They are a pest. So we have decided. Notorious for spreading disease in crops. What they do is by using this kind of they have this thing on their tail. It is called an anal stylus, which is what you kind of saw flipping back and forth, or as the researcher Bamla termed, a butt flicker.
Really?
when they're ready to urinate. Well, he's a technology student, I guess. I don't know. He's not, Anyway,
This is not you. This term is probably not used in the research literature, but is yeah. Got it.
When the sharpshooter is ready to urinate, the anal stylus rotates from a neutral position backwards, kind of on point to make room. The insect squeezes out the liquid in this droplet. The droplet forms, grows gradually. The stylus remains at that same angle, like almost like a 90-degree angle. When the droplet approaches its optimal diameter, which somehow this animal has figured this out, the stylus rotates farther back, kind of cocks it 15 degrees, and then it launches the droplet. The stylus can accelerate more than 40 Gs, which, for comparison, is 10 times faster than the fastest sports cars.
That's a lot of force. What's going into that?
Saad Bamla was very interested in this and wanted to now look at the fluid dynamics of excretion, which has not been studied too much. We kind of take pee for granted. But so he wanted to use computational fluid dynamics and biophysical experiments to study the fluidic, energetic, and biomechanical principles of excretion to reveal how an insect smaller than the tip of a pinky finger performs a feat of physical and bioengineering super propulsion.
But so it makes it's the push that propels it that fast, right?
So I'm so glad you.
It's not just
not
the flick.
It's not just the flick. So they used high-speed videos and microscopy to observe what was happening on their tail. They measured the speed of the stylus movement, which they measured the speed of the water, which was 40 Gs, right? They measured the speed of the anal stylus movement, compared it to the speed of the droplets. But the droplets were faster than the stylus that flicked them. How is this possible? They expected the droplets would move at the same speed because it's just pushing it, right?
Yeah, flicking.
But the droplets were 1.4 times faster than the stylus itself, which means the presence of super propulsion, which is actually a specific scientific term, which is a principle previously only shown in synthetic systems, in which an elastic projectile receives an energy boost when its launch timing matches the projectile timing. And so they liken that to a diver timing their jump off a springboard at the exact right moment.
Oh, fascinating.
So they found that the stylus compressed the droplets, which stored energy due to surface tension. And so at this tiny scale, when the water droplets are launched, the energy stored from surface tension, if timed just right, propels them even faster. So,
wow.
So
it's timing.
It's timing. It's all about timing. It's all about water tension. It's all about super propulsion. So this is crazy enough. But now let me remind you, this is an insect that's doing this. Why? So that was the next step of the study. Why would the sharpshooter need to super propulse its urine? They only eat plant.
It doesn't like it.
They only eat sap. It's nutrient deficient. It only has water and trace minerals. They drink up to 300 times their body weight in sap per day. And they have to therefore constantly excrete fluid waste, which is 99% water.
And it can be tracked by predators.
Not exactly.
Okay. Or it could be if it weren't being
that's a great conclusion, though. I mean, that would be a great conclusion because
okay, so
if it's trackable, you want it nowhere near you.
I
actually don't know that. That is possible. Okay, that's totally possible. You're right. This did not study that, so that could be part of it. What they did look was at micro CT scans of the sharpshooters. They used that to calculate the pressure required for them to push the fluid through their anal canal, which is like the act of peeing, right? And determining how much energy would be required to do that. And so they think that this is actually an energy conservation strategy.
Wow.
Since they have this constant fluid dynamic challenge due to their teeny tiny size, their energy constraints, the poor energy value of the food that they eat, and the fact that urinating normally would cause a deficiency in energy. So the droplets are the most efficient way, but then they have to get rid of them. So
let's do it in a nice little bouncy way. I'll pretend my pee is what's his name? Brian Boitano. No. Greg Louganis. No.
Wait. Are you trying to think of
Never mind. Diving people. Diving people. People
Michael
who dive.
Phelps didn't swim. He was
a swimmer.
Yeah. Anyway.
Divers.
Yeah.
Greg Louganis. That's a sad story. Anyway, moving on.
Yes. So anyway, this whole concept of the sharpshooters and their urine has applications for ecology and population dynamics. Ecology because, as Kiki mentioned at the very beginning, they are a pest. So this could provide insights on how you could kind of get rid of these guys, or how disease is moving through crops from them if they have these fluid dynamics. It's kind of a stretch, but it also has insights on how to design systems that overcome adhesion and viscosity. And a perfect example of that is if you have a smartwatch and you hit the liquid eject button. When you do that, it uses the speaker vibrations to repel water from the device. So it could actually potentially use this kind of fluid dynamic study to enhance water ejection technology, which is very cool.
There's definitely some industrial engineering application waiting for this somewhere.
Oh, for sure. Yeah.
It's going to be something very obscure that just completely revolutionizes some microfluidic transfer devices.
I had absolutely no idea there was a liquid ejection button on Apple Watches. Is that what you said?
There is. Yeah.
Oh, now you tell me.
It's true. I do it whenever I go swimming. You hit the water button, and then you hit the water button when you're done. It goes psh, 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 psh. It's very cool.
We need those for kids' ears.
Yes, that would be nice. You to understand the fluid dynamic.
No more swimmer's ear. Let's get past it.
Anyway, super propulsion.
Superpowers. What I'm wondering though, I mean, this is fluid dynamics. Our atmosphere is a fluid. Can we use this to superpower diving boards that launch rockets into space? Blair, your face right now.
What?
Yeah, why not?
Sure.
Why not?
Flick a spaceship like a ball of urine.
Yeah.
You said these little balls of urine hit 40 Gs.
Yeah,
that's substantial. Right. And if we can use any kind of fluid dynamic technology that maybe does not involve human beings in the crafts, but helps get us to launch things off the planet using less toxic fuels.
Well, you'd have to launch from a trampoline, right? You have to get the momentum going.
Sometimes you just got to throw ideas out there, see where they go.
Taking all the kids down to Cape Canaveral to watch the giant anal stylus.
Yes.
No, it's a rocket launch.
Flicker.
It's the rocket flicker. Thank
Oh
you
my
very much.
goodness gracious, I don't know.
It's the super powered rocket flicker. Okay, Blair, you wanted to talk about
Yes.
hormones now?
Oh yes. It's a pretty simple story. Red-bellied lemurs, they find each other, they fall in love, they're monogamous, they live together in close family units. Offspring stay with them for about 3 to 4 years, and they go off on their own. But as they do that, they're one of the mammals that you can look at that have a really significant parental care from the father as well as the mother when the father is around for the whole pregnancy. And the pregnancy for a red-bellied lemur is about 126 days, or a little over 4 months. And in the 3rd trimester of their pregnancy, males see a 4-fold increase in estradiol, which estradiol is an estrogen steroid hormone and major female sex hormone that is usually associated with an increase in maternal sensitivity and responsiveness. So this is something that hasn't really been looked at before. There's been lots of studies on changes in hormones in females and corresponding changes in hormones in males. There have been studies on, where is it? Different hormones, though. It's cortisol, oxytocin, prolactin, androgen. They've all been shown to change significantly in humans, monkeys, certain rodents in response to partner pregnancies. But estradiol has never been looked at, mainly because it's historically a female hormone. So it's just kind of like not looked at. But this is the kind of the first time this has been seen. This was looked at in feces in lemur poop. So it's something, hormones are usually really easy to test in fresh poop. So if you can get there when it's still warm, you can find some really good hormone readings. And so the kind of suspicion is that this hormonal shift is nature's way of preparing male lemurs for fatherhood. There have been some experimental lab studies with rodents that shows that estradiol impacts parental care, and it's important specifically for maternal care. But so now this is definitely one of those, like, more studies are needed. Now's the time to really look at estradiol. They weren't able to find, because it wasn't like a controlled experiment. They were just picking up poop from the forest. They weren't able to find a direct link between estradiol levels and specific parenting behaviors, like holding, carrying, grooming, playing, huddling. But they do think, based on the fact that it is so clearly in the 3rd trimester, just this huge spike, that it does essentially have to do with preparing them for parenting in general. It's also interesting that they don't drop immediately after birth. The estradiol levels stay high until they're weaned, which isn't just a timing thing, because they can give birth at different times of year, but they always wean in the spring, which is when there's the most food, so they don't need the milk as much. So the fact that the estradiol levels plummet when weaning happens, regardless of how old the baby is, means it definitely is related to the presence of the baby.
Right. But this might be different in species that don't have paternal care, right?
Oh, absolutely.
So
yeah,
we
that
look
would
at
be
it.
the expectation.
Yeah.
It's all about that new baby smell.
Yes. Yes. Yes, but also, like, I don't know, I could ask both of you, Justin, do you think you acted differently when your significant other was in their 3rd trimester? Do you think you were being impacted by hormones?
Yeah, but I don't think they were mine.
But see, this study is saying her hormones are affecting your hormones.
Yeah. That I had no idea.
Yeah.
Yeah.
It's interesting. Kiki, do you think you saw any changes in Marshall?
It's a long time back now.
Yeah.
Yeah. I mean, it's interesting. It's an interesting question, though, because it's it all becomes so real when you get into your third trimester.
Right.
You know, baby's bigger. Like things are really starting to get into preparation time, and so there are a lot of social pressures. And so I don't, I would love to say that, yeah, I saw these things, and oh, how interesting that they could have been, influenced by my hormones. But it's, it's so
I think
complex with people.
Yes, there's
it's
lots of
just-
variables,
right?
it's
Yeah.
just
so many
another
variables.
example, though, honestly, of men's health being understudied. Because nobody's paying any attention to male biology.
I'm just gonna go to
during
sleep on my
pregnancy.
microphone here right now.
Oh no. Well, so it's interesting you say that, Justin, because as silly as that is,
Yeah.
that is kind of the point of this study,
Yeah.
is that there's so much focus in pregnancy on the individual carrying that there's not a lot of study, if there's a monogamous mate in the picture, on that individual. And so obviously the most important thing is to pay attention to the oven, like the individual carrying the baby. But there is this other piece that's really interesting. And so it might be worth looking at in many different mammal species that are monogamous. You specifically have to look at the monogamous ones, or not even monogamous, but ones where the males partake in parental care.
Parental care. Yeah.
Yeah.
Yeah. It Yeah, I think it's a very interesting point to bring up, that we have segregated the research studies that we do based on the roles that we expect particular sexes to partake in during particular social activities. Reproduction. Okay, women, we're going to study all your female sex hormones. We're not going to look at them in men or even check to see if they exist. Like if you're being influenced or what's happening, all sorts of things that we've seen over and over again. It just goes to show there's more work.
To be fair, what the scientists were assuming is that, like you said, Blair, it's all about the oven, which is what you learn as a man who has somebody who is about to be giving birth, is that it has nothing has anything to do with you.
But why?
And your hormones or your moods or your needs and everything doesn't matter anymore.
But it's not just
logical.
And we've learned it's not just oxytocin now. Oxytocin doesn't really play as much of a role in all this bonding bonding, yes, but it's all very dopamine and there's a lot of serotonin and other stuff involved here. But that's the question, right? Social animals, monogamous social animals, the males are taking care. And if there are no cues to tell them to do that, then why are they doing that?
Right. Which also leads me to my other question about, like, if this same thing is happening in humans.
Yeah.
And there is a hormone imbalance in one of the individuals in this relationship, is that impacting the other individual? Is that impacting parental care given to that baby? Similarly, there's lots of research on postpartum depression, and if that has to do with hormone regulation, and if that is happening, is that impacting the other individual and the care that is being given to that child? Like, it could potentially have pretty big impacts.
And it could lead to a screening test where it's like, okay, well, before we even talk about dating, I need to see your capacity to produce hormone profile.
Yeah. It's like Gattaca for hormones.
Biologically, you're predisposed to fatherhood.
Well, I just think about, like, if you have a problem producing estradiol as a man, okay, and you are having trouble bonding with your baby, that is something that you could look at medically. There could be a hormonal cause for that.
Maybe could be
better parenting through chemistry.
It's part of it, man. It can be part of it. Hormones are a big part of our lives.
Absolutely.
I think it's so interesting how much they can impact us.
Oh my goodness. You think you have free will, but really you are just a movable sack of hormones.
Hormones and
Influencing.
bacteria and viruses.
Yeah. The hormones, the bacteria and the viruses, they're affecting your hormones too. Those are
Yeah.
affecting your neurons. That's all making
Yeah.
your behavior change. So really, it's all about the hormones, everybody.
And that is what's going to separate us from the organoid AI
Brain computers.
brain computer
Right.
overlords.
Ah! Oh my goodness. This is This Week in Science. Justin, what do you want to talk about?
So I'm going to wrap up the show today, or my portion of it, with 2 studies about ancient genetics in European current modern humans. Both of these are published in Nature, Ecology, and Evolution. Both of these are from the Max Planck Institute of Evolutionary Anthropology. And the first, they talk about how, well, there was the first current modern humans to spread across Eurasia, started over 45,000 years ago. Previous research has shown that those first modern humans that arrived did not contribute to later populations, which is weird if you think about it. As an aside to what the story is going to talk about, which is weird if you think about it, because this is the group that would have had the most, assumingly most contact with Neanderthals, yet isn't the ancestor of people with Neanderthal DNA. Researchers, however, did focus on the people who lived in Europe between 35,000 and as recent as 5,000 years ago. This is the group that would have been there right before the Bronze Age, populating out of the Caucasus, which is where that mapping system said there would be a really good spot to be looking in northern Iran. So they looked at genomes of people living during the last glacial maximum in there as well. The super coldest part of the Ice Age, around 25,000 years ago. The team analyzed the genomes of 356 prehistoric hunter-gatherers from different archaeological cultures, including new data from 116 individuals across 14 different European and Central Asian countries. Research team found that populations from different regions associated with the Gravidian culture, which has been archaeologically identified by their artifacts. They all use, even though they're kind of spread out over a large region, you can tell they're part of the same culture because they use similar weapons, produce similar portable artworks, have similar techniques within their tool manufacturing. And it was very widespread across the European continent between 32,000 and 24,000 years ago. But kind of like you were talking about earlier, Kiki, until you get genomic, as long as you're talking about morphologies or artifact culture, you may be missing part of the story. Turns out right here, 2 groups, one from the sort of southwestern Europe, today's France and Spain, and the other group of Gravidians from Italy into the Czech Republic, not related. Very disparate, unrelated populations, despite the appearance of cohesion from the artifact record. Turns out the gene pool in Western Gravidian populations was found continuously for about 20,000 years, sitting out the Ice Age in the comfort of southwestern Europe. Meanwhile, the Gravidian population on the Italian peninsula was no longer detectable after the Ice Age, as people with a different gene pool, the Epigravidians, settled all of Italy and Sicily. So they got replaced. Then these Epigravidians, these sort of invading inhabitants of the Italian peninsula, spread across the rest of Europe about 14,000 years ago, replacing the populations of Gravidian cultures there. Study also reports that there is no genetic exchange between hunter-gatherer populations in Western or Eastern or Central Europe for more than 6,000 year span, and only began detected about 8,000 years ago that they started to intermingle. And once they did start to intermingle again, they would have had very distinct ancestries and even appearances. in the second study. So that's kind of what we kind of think of as, humans coming in and eventually populating Europe, and then Europeans today, completely unrelated. The Europeans of today are more descended from much more recent arrivals. And then the second study, which is probably part of the overall analysis there, researchers analyzing human DNA from several archaeological sites in southern Spain discovered a 23,000-year-old individual from Cueva del Malamurezo, which is near Granada. And it is the oldest DNA from a current modern human ever recovered in Spain. The researchers were able to genetically link that individual with a 35,000-year-old individual previously found in Belgium, making it part of one of the earliest lineages that settled Eurasia around 45,000-ish years ago. The study also includes a number of more recent individuals from the Neolithic, 7,000 back to 17,000 years ago, time period when farmers started to first arrive in Europe from the Near East. Found that the genetic heritage of Paleolithic hunter-gatherers was still detectable in these early farmers, so that the last hunter-gatherers of Europe did mingle with the first farmers who arrived.
That's fascinating.
Findings further suggest, well, I'm suggesting, but based on this, that people still hadn't invented boats. Boats is one of those things that we have a hard time decisively saying when they started, because if they're made out of wood and they were kept coastal, it's very hard for wood to stand the archaeological tests of time. So we might have just lost evidence of it. There is DNA that had been recovered previously from 14,000-year-old North Africans in, I guess, what would be Morocco, the 13 kilometers across the sea from southern Spain. And there were no signals of the North African DNA in the Iberian genetic cultures, and none going the other way either. So even though you might have been able to see a shoreline on a clear day off there in the distance, people walked all the way around through that darn southern Caspian Sea corridor, up through the Caucasus, back down into the... It took a long way.
It's gonna take me a while.
There's sea monsters. You can't go in there.
Yeah. Haven't you seen the pictures my friend drew of a thing he heard somebody describe when they were drinking? I'm not going in the water.
No.
They'll eat you up.
I'm still puzzled of how the Neanderthal signal doesn't come from the folks that were the most likely to have encountered it, and a couple times over, which then means that despite the early interactions that we could have been having with Neanderthals, the ones that led to the current DNA admixture must have happened either much more recently, or they must have taken place somewhere like the southern Caspian Sea corridor before the new waves entered Europe.
Right. Yeah, so all of these stories that you're reporting on tonight, they all kind of tie in together into this potentially new way of looking at that.
There's a potential narrative there that could make sense. It just, of course, needs, what's that thing? More research. That's the thing that's needed.
Oh, I'm so glad we're going to need scientists for a very long time. We need scientists and all the scientisting. All right. I have a couple of stories to close us out. Are you done with your stories, Justin?
Yeah. That's
fantastic. I have a story out of Caltech, Nature Communications. Researchers in the lab of John O'Doherty, Caltech's Fletcher Jones Professor of Decision Neuroscience. They have been trying to figure out what it is about people's brains that decides what art you like or don't. Do you like abstract art?
Yes.
Do you like Renaissance? Do you like realistic art?
Not as much as the abstract.
Right. So everyone has their personal differences and the things that they like aesthetically. Food, making a food decision, now that's something that can be broken down into components that make sense, related to nutrition, related to energy levels, related, there are all sorts of aspects that food choice and the brain kind of makes sense. Those decisions seem a little bit easier to break apart. But what about liking or not liking particular kinds of art? So these researchers in their study in Nature Communications had a whole bunch of people lie in fMRI machines and look at art while they answered questions like, did you like this? And then they, on a scale of 1 to 3, said, no, absolutely not. Or yes, very, very, very much. I like it very much.
I like that scale. I like a 1 to 3 scale.
It's not a big scale. It's just very...
But that's, that's a 1 to 3 is a perfect scale because it's either yes, maybe, no. And that's all your answers should only, it should be no 1 to 10, 1 to 100. It doesn't make, it's too much. There's not enough nuance in a human being for that big of a scale. 1 to 3 is perfect.
Who needs all of this nuance? Well, okay. Visual system, we know, recognizes images based on lines, on contours. It looks at these things like low-level aspects, and then they get built together to create higher-level context or features. And so you have to start thinking about at what point in this train of your visual system, breaking everything apart into its component little parts and then putting it back together again, at what point does the I like it or I don't like it happen? And so by looking in the fMRI machines, they were able to start taking a look at the various parts of the brain that were responding to the images and that coordinated with the particular liking or not liking, the art rating task, which I really like the creativity of their task. It's called ART, A-R-T, art rating task. Each participant, there weren't very many of them, but each participant completed about 20 scan sessions over 4 separate days. So each individual was like a repetition in themselves. So they had a lower number of people involved in the study, but each person did a lot of repetitions. And they were able to create a computer model that correlated changes in different parts of the brain, like the prefrontal cortex and other areas that are higher level cognitive areas that are responsible for decision making and our cognitive control. And they were able to see how the lower levels of the visual system eventually affected a whole bunch of other areas of the brain and determined this subjective value. Now, at this point in time, they cannot look at your brain while you're looking at something and predict whether or not you're going to like it. Because each brain was one person's brain. So their model works in terms of the breaking apart of things and putting them back together, but they have not yet developed the predictive aspect. So we're not at the point. I was expecting you to say that. Yeah, we are not at the point yet where this research is going to be, the AI that's in your Siri or your Google or whatever, whatever's watching you while you're watching things and doing more than just responding to your clicks. They're not reading your brains yet to be able to deliver you specifically, the next Jane Austen novel readaptation.
But they are following our purchasing preferences and the things that we like online and the things that we share. And so there's still a pretty good predictive power going on in other ways.
What I would
Absolutely.
What I would love to see is for this same study in the future to include those horrible little artwork descriptor post-it note things that are near paintings
that tell you what you're supposed to think about. What
exactly?
This is what the artist.
How that affects the actual appreciation.
This plain white canvas is supposed to evoke sadness.
Yeah, because I think those things should be illegal. I really do.
Because it's telling you how to feel about art. Yeah. Is that why?
How dare you tell me
Don't tell me how to feel.
View this painting with my brain. Get out of the way. Stay away. Never read those. And if you see one in a museum, complain.
I like when museums have them where they don't explain what the painting means, but they maybe explain something about the artist or about the context in which the painting was made. I like that. I think that's interesting.
Oh, that's tough. Oh, this was a painting while the artist was living in isolation in a cabin, recovering from a bad relationship. Stop it. Stop. If I couldn't tell that from the painting, that's not what the painting's about.
Well, I mean, it's a little bit better, a little more context. There's never enough context.
Even the title
Walden
of the
Pond.
painting,
the dude was supposed to be like by himself, isolated on Walden Pond, writing, but his like mom and sister were coming and bringing him food and doing everything for him. So he didn't actually have to struggle.
Even
there's
the
a whole different interpretation once you learn that.
Of a painting, I find obstruct. Like if it was like covered with a flap where you're like, I want to know the name of this painting after I've like looked at it so that I can reference it. Absolutely. The painting could have a title, but you shouldn't be able to see
the artist's name
until after you've looked at the painting for a little while.
Well, but also, like modern art, the painting, the title is half of the art. Like when you have a urinal and it's called water fountain. That's the whole point of the art.
My favorite part of my trip.
The point of yes, sure.
Yeah, my favorite part of my family's trip to Europe this past summer, was going to the Modern Art Museum in Paris and having my child walk stubbornly through the entire museum going, I hate this. This is dumb. Why do they put this in a museum? What is this? This is white. I could do that. What is that?
Yep. Listen, Kai and I would get along in that museum for sure.
Oh, I was like, I'm going to keep my distance from my child here. I'm going to go enjoy some art. Oh my goodness. Last study for the night. Now we're going to talk about Carl Disseroth. Carl Disseroth, you might recognize his name because he has been involved in the development of optogenetics, which is the technology that allows us to be able to use genetic modification to be able to insert genes into neurons, into cells that respond to light. Usually blue light. The blue light's really awesome because like if you're dealing with cells on the very top of a thin-skulled animal's head, you can cause behavioral changes in the brains of animals. And it's really amazing. We've talked about the problems with how that would not necessarily work in people very well because blue light doesn't go very deep and like, also the gene modification and all that kind of stuff. But new study. Researchers, they have in the Disseroth lab, they've just published in Nature their study that found, you're going to be so surprised about this, An elevated heart rate can cause anxious behaviors in mice.
What?
But only in risky circumstances. So
Wait,
if it's like the
causal.
mouse is in a place like its running wheel where it's running and running and running and its heart rate is normally elevated, if they optogenetically made the heart rate elevated in that place where the heart rate is normally elevated, there were no anxiety behaviors because that's normal. But if they made the heart rate change out in the open where predators could be, then anxiety in the mice. New part.
Okay.
Yeah. So it's interesting. So it's
Huh.
context related, which I
Yeah.
think is also we're looking. I mean, this is not. I have no science backing this up. I'm putting multiple scientific things that I've read together in an analysis, just in this idea that we talk about exercise is good for people who have anxiety and depression. That getting outside, that doing, and these things elevate your heart rate normally. So, is there something to be said about the fact that you are going about behaviors that elevate your heart rate to normalize the feeling of elevated heart rate with respect to certain circumstances? Okay, that said, this new technique, they've been adapting their channel rhodopsins that they use. One is sensitive to red light. Red light can go more deeply into tissues. And so that is how they were, in this particular circumstance, able to change the heart rate of the mice. They used channel rhodopsins, which they call chromine, CHRMINE, and pretty much use the same optogenetic techniques that they've used previously for the brain, but they have been able to develop basically an optical pacemaker of sorts where they could turn on the light. Red light makes the heart cells go, "Ah!" and the heart would pump more quickly and actively, or they would let it go down and it would go back to a resting heart rate. Additionally, they changed the activity in different brain regions that they know are related to anxiety in these animals to determine what was happening in particular situations. But yeah, what they've determined is that the ability to be able to turn off some of the brain cells and to stimulate the heart in certain situations, they were able to see what areas of the brain are potentially important when responding to anxious or fearful information versus just physiological information.
But isn't it the right? But the physiological information in this, the increased heart rate, then triggered that response. I wonder if the mice are like, oh, God, hey, I think I'm having an anxiety attack, and that made them nervous. I had a big day planned, but I'm gonna have to.
I don't know about that.
I don't know if I'm gonna go through. I might have to cancel some of my. I won't be there at the cheesemongers today. I'm gonna have to.
The cheesemongers.
Wherever mice go, that's where I'd go if I was a mouse. I'd go over to the cheesemongers place. That's where all the good mouse action's taking place.
Yeah, so
interesting. So there's the feedback loop there.
Yes. Yeah, there's a feedback loop.
You would normally think that heart rate is driven by the anxiety, not the.
But it could be the other way around as well. The heart rate itself is a driver of anxiety. And people who have had tachycardia, atrial fibrillation, who have heart disorders that do lead to changes in heart rate that are unexpected, you do feel these anxious symptoms. And very often you go, I'm having a panic attack. And then, you go to the doctor, they look at your heart and they go, oh no, you've got a little heart murmur going on. Or you have this atrial fibrillation. So maybe you shouldn't be taking that medication. Maybe you should take this one instead. And, lay off the kava for a bit, whatever.
Yeah, I was gonna say, like, people, if you have anxiety, they tell you to cut out on the caffeine.
Yes.
So I wonder if that also is about this, right? The caffeine gives you a heightened heart rate, or it can if you have too much of it.
Right, but it's it's caffeine. It's affecting all sorts of things at the same time. So I don't know. But the techniques being used are really fascinating. It's in Justin, what you were saying there. It's the feedback loop. It's not just brain to heart. It's heart to brain as well, body to brain. So there's these these feedbacks are loops. They're not just one direction. I mean, that was a bad band from, ages ago, but anyway.
It's that whole phrase is, I feel like, belongs on a t-shirt thing. The feedbacks are loops. It's not just one direction. I love it. Put that on the wall of a museum.
Put it up there. Interesting stuff going on. Non-invasive, quote unquote, optical pacemaker. Like maybe someday we'll be using these channel rhodopsins with red light, little flashlights stuck to your chest to manage heart rate. Maybe the optogenetics technology is going to take us very interesting places.
Have they applied it to the organoids there at the lab? It's the same lab.
I know. I don't know. Probably.
Hey, did you see what we did with the mice? Let's see if we can control the behavior of our organoid computer.
Let's make these brain organoids we're teaching to learn anxious.
Let's connect them to a heart.
No.
Make an anxious. Oh gosh, then you've got an anxious AI. Wait a second. That was in Hitchhiker's Guide to the Galaxy, the robot. Yeah. Mm-hmm. That was horribly depressed robot.
I'm so sorry. Yeah. Depressed robots. Not only Hitchhiker's Guide, they're all over the sci-fi universe. But have we made it to the end of the show?
We've done it.
We might have.
We might have done it. Oh my goodness. I would love to say thank you to everyone for joining us once again for another episode, and give some special shout outs to people who help us every week with the show. Thank you so much, Fada. Fada isn't here. He's doing stand-up improv tonight. So hopefully break a leg. Fada, hope everything went well there. Gord, Arnlor, everyone who helps out in the chat room. Thank you for keeping the chat rooms wonderful places to be. Identity 4, thank you for recording the show. I hope my choice of streaming links didn't mess things up too poorly. And I'd like to thank Rachel for editing everything so nicely. And finally, of course, I can't forget our Patreon sponsors. Thank you to Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velzar, John Ratnaswamy, Carl Kornfeld, Karen Tozy, Chris Wozniak, Dave Bunn, Vigard, Chefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Derek Schmidt, Dan Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky, Luke Paul Runovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Lawrence, Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Rick, Ramus, Phil Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you all for helping to support TWIS. And if you are interested in supporting us on Patreon, head over to twis.org and click on that Patreon link on next week's show.
We will be back broadcasting Wednesday at 8 p.m. Pacific time, and again on Thursday, 5 a.m. Central European time, from our YouTube and Facebook channels, as well as from twis.org/live.
Hey, do you want to listen to us as a podcast, perhaps while you get your heart rate up in a controlled fashion on a treadmill? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twis.org, where you can sign up for our newsletter.
You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be flung at 40 Gs, and we'll never find it.
Nope.
Ever.
It'll be gone. But never mind. You can also find us on the Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes through in the night, please let us know.
We'll be back here again next week, and we hope you'll join us for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.